Welcome back. Today we're going to have a quick discussion on basically, it's going to be a primer about genetics and also kind of a discussion on the way that we talk about this within the hobby. So the first thing that I want to bring up is, so right on the screen here, you'll see a little illustration that I did. Obviously, I am an artiste. So this, I've drawn what you'll see on Facebook and forums and a bunch of other stuff, where somebody will show up and they'll say, listen, this is how inheritance works. If you have an inchy ball python and you breed him, then what happens is you have this double helix, and the double helix cuts in half, and then that's how they go ahead and this is completely wrong. This is completely wrong. This has more in common with cloning. If this was the way that animals bred, then this would be a clone, because nucleotides on either side can only attach, or however you want to say it, bind to complementary nucleotides. So this is completely wrong. This actually has more in common with PCR, which will be what we'll be doing in a future video, because as you know, I bought all this equipment and did the mini PCR experiment and managed to identify lavender albino based on the great work from the ball python genetics project and many researchers. But anyway, so this is completely wrong. If you start seeing somebody talk about double helixes and stuff like that, just completely ignore that. When it comes to animal breeding and when it comes to sexual reproduction, DNA is actually encapsulated inside of chromosomes. So here you'll see I've got it set up vertically. Each chromosome on the left has a complementary chromosome on the right. You can think of chromosomes like big balls of yarn that do contain DNA. They do have that double helix all twirled up in there. But they are convenient little packages. When a cell divides, it completely clones all of its chromosomes to the next cell. When animals sexually reproduce, they only pass down half of those chromosomes. So again, let's go ahead and refer to my amazing drawing here. The thing that I'm using on the right side here is actually it's called Excalidraw, and it's just a fun little program that you can use right in your browser to go ahead and draw stuff, right? So here we've got the chromosomes illustrated. The two wavy lines, of course, means that there's more there. I just didn't draw all of them because I would have been here forever. And then at the very, very bottom, I've gone ahead and included what are known as the sex chromosomes. So we've got good old dad and good old mom here, kind of laid out the same way that we were just talking about, where we have these pairs of chromosomes that you can see. So, dad, his first set of chromosomes, he's got a red one, got a green one, mom's got a blue one, purple one, whatever color that is, I'm not really sure. At the very bottom, you can see the sex chromosomes. So an XY animal is, of course, male, and an XX animal is, of course, female. There's also a lot of back and forth on how exactly banana works. So we'll go ahead and cover that, since we've already got this amazing piece of artistry, this amazing illustration that I've created, right? It's not really that amazing. It's kind of awful. But anyway, just to make that clear. So mom can pass down an X chromosome. Dad can pass down an X chromosome or a Y chromosome. Males will have a Y chromosome. Females will not. In the case of banana, if that gene was located inside the DNA that was inside of this Y chromosome, then dad would only be able to pass down banana to males that receive that Y chromosome. To say that a different way, only animals inheriting that Y chromosome would be able to also gain banana, and they would also be male, because that's kind of just bound together in that same little package. Every now and again, we will have that specific gene. So when we're talking about banana or Corglow, that will jump over to X. You can look this up yourself. This is kind of out of the scope of the video, but every now and again, it'll jump over to X. So what that means is any offspring from mom has an equal 50% chance of receiving an X chromosome that includes that gene. So mom can produce XY, which would be males, and mom can produce XX, which would be females. This is how we get things like super bananas. Sort of kind of out in the grass example too is mourning geckos. Not all reproduction of mourning geckos is just from the females. Every great now and again they throw out a male, and that's how we get diversity within that species. It's really interesting. You can go check this out on your own. That's kind of out of scope for this video, but worth mentioning. So just to follow my little illustration here. Now, dad produces sperm, obviously. Mom produces eggs. And when the two meet, that's how we start to get cells that are eventually going to become an offspring. So inside of the sex cells are only half of the chromosomes from dad and only half of the chromosomes from mom. Now I'll repeat again that these chromosomes have an equal 50% chance. So I've shown here that mom's going to pass down this blue chromosome, but it's just as equal as she passed down the other chromosome. It's an equal 50% chance. The complementary chromosome over there from dad, of course, is green. So we've got some other colors here. There's a yellow one here, which so up here on dad's side, I went ahead and marked 2 of these as this orange color. I thought that would be a great indicator for something like pastel or orange dream or whatever you want. Make it NG, doesn't matter. But so we'll say that dad has 2 chromosomes that include a specific section that codes for super pastel, or I'm sorry, encodes for pastel. And since he's got 2, then he's super pastel, right? So an equal 50% chance of either of these 2 little orange chromosomes passing down, you've got a 100% chance that gene is going to pass down because both chromosomes have that gene in it. So in this case, we've got this little yellow, orange. I'm really bad at colors. We've got this little chromosome here that has pastel in it. So 100% of these offspring are all going to carry pastel regardless. Mom has a similar chromosome, but it is in a different position in the chain. So that means that this could not be super pastel, because super pastel would be a very specific segment of DNA inside that chromosome that coded those cells in a specific way to express that phenotype. So again, that would be the genotype when we're looking at this. The phenotype, of course, would be what we can see. So we know that we'll have a pastel, and we also see here that dad has passed down a Y. So I'll say this again. Dad had an equal 50% chance of passing down an X or a Y, but only XY will be males. XX will be females. Mom has 2 X's, so she has an equal 50% chance to pass down either X, but she's always going to pass down an X. So this is where we get, roughly 50% of these animals should be male or female. There's a little segue here where people argue this is per egg or this is per whatever. Ultimately, I mean, it's all probability. Generally, we see animals that, half the clutch is male, half the clutch is female. Every great now and again, you have a clutch of all females or all males. The same probability for if you somehow manage to have only a single gene Wookiee, let's say, and somehow 100% of the offspring were Wookiee. It's not outside of the realm of possibility, it's just generally improbable. So that's just a quick rundown on probability. Now we're going to put all of our chromosomes together, and we've got Boyo here. So we have somebody for Kratos to yell at in the latest God of... No, wait a minute. That's a different video. So Son here has that little pastel, or he has the chromosome that contains the pastel gene. You can also call these loci or allele. You'll hear words like this kind of slung around, but for right now, I just want to focus on everybody understanding that chromosomes are the inheritable component. It's not the double helix being torn in half or anything like that. But anyway, so Son has inherited these specific chromosomes. When we talk about things like the Ball Python Genetics Project or the DNA testing that I've done in a previous video, anything that Rare Genetics has talked about lately, Justin Kabilka had a really great, I think it was part of Kabilka University, where he talked about, or sorry, Canova, he talked about genetic testing and how it might change the hobby. This is how. So these genes are located in pieces of DNA that are inside of these packages called chromosomes. That's what we're looking for. Those pieces of DNA will be unique inside that chromosome, and so that's how we get genes that we call allelic, because they exist in that same package and they exist in that same area that affects that animal's phenotype, that is, inchy or pastel or whatever. So if we were to talk about something like Candy and albino and how they create Candino, that's because they are related on that same chromosome around that same segment and are actually allelic when it comes to the combination of these chromosomes that are ultimately the animal's DNA. Hopefully that makes a little bit more sense. I don't like to see people saying that the double helix is ripped in half or whatever, because that's literally cloning, and again, has more in common with PCR, which we'll cover later on because of the experiments that we've been doing here lately. So that should also help explain banana too. I know there's a lot of videos that have explained this and a lot of videos that have explained banana, but I thought that we would just go through it again just because I should put videos out on this channel. Now, let's go ahead and talk about a few things that might be hard pills to swallow, might be stuff that people are going to get a little bit upset with, because I can't say anything in a video without upsetting somebody. Ball pythons are very unique. Almost every single gene or phenotype or whatever we want to call it, almost every single one is a simple inheritance or a Mendelian inheritance. When it comes to animals like crested geckos, however, these animals have very few of these traits that are inherited along the Mendelian model. So that, obviously, if you look at something like lily white, then lily white is Mendelian. Half of the babies from a lily white should produce, or half of the babies from a lily white will be lily white. When it comes to exanthic, of course, that's recessive. So you're going to need either two parents that are exanthic, or a visual and a het exanthic, or whatever combination, in order to get a baby that is going to be exanthic. So in the case of something like lily white, automatically guaranteed 50% chance. In the case of something like the exanthics, then you are going to need to, of course, map out your hets versus visuals versus whatever, right? Obviously, if you have two visuals, you're going to have exanthic babies. And a fun fact too, that's actually should be exanthic, but you know, whatever. As long as everybody knows what we're talking about, pronunciation isn't that big of a deal. That also goes for Mendelian or Mendelian or however you want to say it. So the way that crested geckos work are polygenic, right? Not polygenetic. That is incorrect, and you'll know that I already got in trouble where, you know, Mr. Justin Smith from Herp Magazine got really upset, filed a false copyright strike, all this other stuff, because Dr. Travis Wyman got the term completely wrong. Polygenic. 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 3 times. Beetlejuice shows up. The other thing that's very, very hard to swallow, and this is bordering on one of those things that is I wouldn't say it's controversial, but I'm sure it will be controversial. When it comes to things like ethical keepers and certain people that run around in that kind of area, so you'll see videos from the Animals at Home Network, you'll see JTB talk about it, you'll see people like Liam Sinclair talk about how, you know, morphs are bad for the industry and all this other stuff. Here's the deal. Now, when it comes to actual changes in the animal, when it comes to different phenotypes, things that we can observe. Now, phenotypes don't only have to be the color of the animal. It could also be something like defects that cause bad development of the eyes. If you jump over to a few years ago, there was a baby that was actually gene edited, and the goal was to try to make them immune to HIV-1. That was successful, but what was also kind of discovered is that same change also had some correlation to better test scores or something along those lines. So this would be an inadvertent change in the phenotype, not the one that was specifically targeted. Or was it? That's a whole controversial thing too. Of course, if you're in ball pythons, then you already know the same thing applies to something like spider. Even though the visual phenotype is the pattern, we also know that there's a phenotype expressed as wobble. And I'm not going to get into do all spiders wobble. You probably already know my take on this. That is something that was also in an Animals at Home Network podcast where I was at. I'm not a big spider person, but anyway. So when you look at an animal and you decide this animal is obviously flawed because it has morphs, the wild type animal would be genetically superior. This is completely wrong. People died in World War II. We actually had a civil war here in the United States about this, and we continue to have these problems in society where people think that they can, you know, look at something and decide by its skin color whether or not it's genetically superior or not. There's a certain thing called Godwin's Law, which is definitely go look that one up, which should tell you that all of this is completely incorrect. You can't simply look at a snake and say that's a wild type, that one's healthier. That's not exactly how it works. You know, omitting things like spider. Obviously, if you're looking at a spider, you're looking at a normal, go with the normal. But the point being is that snakes in the wild, they don't move very far from where they're born. You can go check out Dave Kosman's latest video. He'll talk about piebald ball pythons that are in a certain region. There have been a few different studies that have talked about snakes that have rebuilt populations from just a few individuals. Now, to be clear, I am not telling you to inbreed your snakes because that's what they do in the wild. I'm not telling you that whatsoever. But the fact of the matter is that snakes don't go very far, and so in the wild, it is likely that they are actually doing a little bit of mixy-mixy, right? Now, in the wild, when it comes to evolution, a lot of people still have this concept, which, again, people died in World War II for you not to think this, but people have this concept that evolution is like a triangle, and eventually you will have the blonde-haired, blue-eyed super soldier. And that is completely wrong. It's completely wrong. Evolution functions, in fact, the exact opposite way. So we start with an animal, and that diverges. It does not converge, it diverges into more diverse animals. Now, this also works for things like bacteria, which has been proven by scientists, and it also works for things like grasses. What they saw was we didn't end up with a single bacteria that was extremely efficient and the perfect creature or whatever, and we didn't have a grass that was extremely efficient. What happened instead is we had some bacteria that were more efficient at eating food, some bacteria that produced more waste, some bacteria that might have been faster, some bacteria that may have been slower. And that's how we get that up-down, or that upside-down triangle. So you get a diverse number of organisms through evolution. That is literally evolution's job. Especially when we talk about things like sexual reproduction, the idea is to have as many diverse animals as possible. The grasses that scientists produce, some use more water, some use less, some were slightly different color, etc., etc., etc. Again, these are things you should be able to trivially find with any amount of just a little bit of research. We want more diverse animals, right? So when we sit there and say things like, well, captive animals can't be healthy because, you know, there's just no fresh blood or anything like that, there's a question there on if our captive animals, they can't be any more or less evolved than the wild animals. This is something that, you know, go take a trot down reading some books by Richard Dawkins. It will make your life a little bit better when talking about this stuff. But when we go down that path, the animals that we have in captivity are no more or less evolved than the animals that we have that are not in captivity, especially when it comes to animals like ball pythons, where people are constantly interacting with these animals. Again, go check out Dave Kaufman's video. He did an amazing job of showing off these animals in their wild habitat and how people were relocating these animals, pulling them across, moving them to farms, moving them to different places. They have their own captive breeding programs down there, and of course they also have wild populations down there. He did notice that in certain regions, and this again, no genetic testing or anything like that, but just from phenotype, he seemed to believe that animals from different regions had different colors. And that's not much different than something like a king snake here in the US. California kings come in all sorts of different morphs and colors and everything else. And the morphs can be wildly occurring. Again, ball pythons were found to be, you know, piebald. There was a whole region of them from what somebody said in Dave Kaufman's video. But also we will see things like rat snakes that produce leucistic offspring. Now, leucism, or leucism, or however you want to say it, again, these are terms that as long as everybody knows what we're talking about, it's fine. So somehow two animals that carried this same this gene that's set in the same place somehow got together and produced a wild leucistic animal. This happens. This is what's supposed to happen. This is what's supposed to happen with sexual reproduction. There is some science out there that says that if we look at genes and we assume that there should be a random pattern of which genes are damaged or which genes change, that is, so when you look at something like a lavender albino, that is a knockout. Part of that DNA literally got broken and then put back together, missing a piece. What caused that? Solar rays, radiation, who knows? Whatever caused it, that's how we got lavender albino. So that is obviously an error in DNA copying. But this is something that is, as far as we can tell in our natural world, happens all of the time. And it should happen all of the time in order to give us an even more diverse number of animals, a diverse number of organisms, I should say. So to have somebody sit there and say something like, that snake is healthier just because it's wild type and I like wild types more, judging an animal based only on its skin color is not only a cultural problem, but something that has been busted science for a long time. Now, when it comes to humans interacting with animals, of course, we may prefer animals that have a certain skin color and things like that. And that's where terms like husbandry come in. But overall, just looking at something and deciding that it's not, it's somehow genetically inferior just because it has pastel or albinism inside of it, that is fundamentally flawed. People died in World War II for you not to believe this. Sorry for people that are going to find that difficult to talk about, but that is exactly where we stand with this. So, just to recap, so somebody doesn't get confused, I'm not telling you to inbreed your animals. Does it happen in the wild? Yeah, of course it happens in the wild. Is that the reason that morphs emerge? Probably not. Morphs, of course, recessives would need some population that were carrying these genes in order to produce this visual animal. However, when it comes to things like dominance and stuff like that, just, again, space rays, whatever it is, right? Comes in, knocks something out, and then now all of a sudden you have a different colored animal. Is this bad? Is this disease causing? Not that we know of. Again, we know with some morphs, so again, spider, jag, there's the whatever it is, yellow mice, whatever it is, that all have some sort of additional phenotype. That is, they wobble, they produce lethal supers, something is not right with them. So we know in that instance that morph influences more than just the pattern. However, in general, we don't have that much of an indication of the way that these animals are affected by these morphs. Even when it comes to albinism, we know that there are albino humans, and albino humans tend to have, they have some trouble with their eyes because the pigments do not form in their eyes. But when it comes to snakes, here's a quick fun fact. Snakes have actually evolved eyes twice. Twice. Do we know that snakes are primarily using their eyes as a way of navigating and hunting? Well, when it comes to ball pythons, we know that they don't. Ball pythons, of course, have heat pits, and if you've ever fed a ball python before, you'll know that it's not, looking at you, hunting down the mouse. You'll know that it locks on with its tongue, its sense of smell, and those heat pits. Those heat pits are actually really high resolution. We have studies that talk about the actual structure of ball python eyes that have evidence that support that they're not hunting with their eyes. They're not using any sort of UV light or anything like that, to hunt down these animals. They may use that as a hint to locate rats or mates or something like that based on scent trails. Well, sorry, not scent trails, but chemical trails such as urine, which would actually light up under UV. They may be using that as a hint of where to go, but ultimately when they are hunting down their prey, they probably are not using their eyes or relying on their eyes as much as we think. Of course, people have blind snakes, blind boas, blind pythons that are just fine, and you don't have to shove the food in their mouth. They can still locate it, primarily because of those heat pits. So hopefully this, of course, was a bit of a long-winded video. The short version, of course, is that, we don't want to talk about the double helix splitting in half because that's not really helpful and that's not really what we're looking for. You should really think about it in packages of DNA, which are called chromosomes. And each of those chromosomes has an equal 50% chance to be passed down. In the case of something like banana, if it's attached to daddy's Y, because males are XY, females are XX, then that will only produce males because that banana is attached to that Y gene, except in very rare circumstances where it somehow jumps over to X. Since females are XX and males are XY, if you do have a female banana, then that means it's attached to one of her X's. And since there is an X chromosome inside males and an X chromosome inside females, she can produce either sex. When it comes to ball pythons inside captivity, are they less healthy than ones in the wild? Very, very questionable. They are simply different than ones in the wild. When it comes to discussing evolution, is there, some wild event that's going to lead everybody to converge to be the super snake or the ubersnek or the blonde-haired, blue-eyed soldier? Absolutely not. Evolution works in the opposite manner, where it produces more diverse organisms, and that includes organisms that may have copy errors in their DNA and where we get things like lavender albino. Next time, I promise to talk more about the mini PCR experiments. That has been going great. Unfortunately, there are a couple hoops to jump through before this is something that somebody can just order and do at home. There are, of course, companies working on this. Check out Rare Genetics Incorporated. Check out the other experiments that Mini PCR has that you can go do in a classroom. And of course, if you're particularly enterprising, you can do this right at home. It's something I did at home. I'm a comp sci major, not a biologist, but it's something that you can do as well, I'm sure. So, see you next time.